Hello and good evening, everyone. I hope my voice is loud and clear. So welcome to another live session on Eden2. Today's live session is exclusively for our second year BDS students. Their university examinations are fast approaching and Eden2 has come up with a revision series to help them in their studies. Okay, so we are already completed the general pathology session and I hope it was useful. Now, today we are mainly going to focus on some important microbiology topics. All right. Okay. So, microbiology, like I had told you in the last session, both general pathology and microbiology are equally important. And there are some overlapping topics between general pathology and microbiology. Comparatively, okay, compared to general pathology, this microbiology is a little bit more vast, and questions that are related to bacteria and virus can be frequently asked. And when I went through the last like 10 to 15 years of the university question papers, they are frequently repeated also. All right. So if you have an idea about which are the like commonly asked bacteria, virus, or fungus, or the other questions that are related to immunology, then it will be much easier for you. So in today's session, I had included that also. Towards the end of the session, we'll be discussing like which bacteria, virus, and fungi you should definitely study before going to attend your university examination. Okay, so towards the end of the session, I had included that also. We'll discuss that also. And yes, let's start discussing the questions. Okay. So, like I had told, lots of essay questions were asked from bacteriology. Okay. Mycobacteria, like Clostridium tetani and Conibacterium diphtheria, and Staphylococcus and Streptococcus. These are all like frequently repeated essay questions from bacteriology. Now, today we are going to discuss such an important bacteria, which is Streptococci. Okay. This Streptococci is important because it is one of the causative agents for the most common oral diseases. Okay, that is dental caries. Okay, so one of the causative agent for dental caries is Streptococcus mutans. So yes, Streptococci is important. All right. So if they are asking a question on Streptococci, the question will be usually like this: classify Streptococci and describe the pathogenesis and lab diagnosis of streptococcal pharyngitis. Or they can ask any other diseases also. Okay, tonsillitis or erysipelas or impetigo or any other skin condition or any other disease they can ask. You should know the pathogenesis and lab diagnosis of streptococcus. All right. And then other than that, the third part of the question will be usually like add a note on the non-suppurative streptococcal infections. Very important and very frequently asked. It can be asked alone as a short note also. Non-suppurative streptococcal infections. Okay, we'll be discussing that also in detail today. And coming to short essays, they can ask short essays like anaerobic streptococci. Okay, streptococci is not including just the aerobic bacteria, but it also has the anaerobic types. So yes, anaerobic streptococci and the diseases caused by them was asked as a short essay and short note question. All right, and then other than that, when I went through the recent January 2025 question paper, there was a question on malarial parasites. Okay, malarial parasites, and this question was like frequently repeated in over the years also. So yes, malarial parasites also we'll be discussing in detail along with its laboratory diagnosis of the falciparum and the vivax species also. All right, then other than that, the culture media, inclusion bodies, and Vincent's angina, every other short note also we will discuss in detail. Due to time restrictions, maybe like we'll not be able to discuss all the questions, but yes, we'll be focusing on the most important ones and which all questions you should study definitely before going to attend your university examinations. I had included that also in today's session. So please watch that also. All right. Okay. So starting with streptococcus. Okay. So the streptos. Okay. Streptococcus, you can like divide the this as strep, strepto, and coccus. Okay. So the streptos means twisted or coiled, and coccus means berry shaped or spherical in nature. Okay, so these streptococcus are nothing but they are the gram positive cocci in chains or pairs. Okay, so this is the typical appearance of streptococci under the microscope. You will be able to see such small spherical berry like chains. Okay, berry like chains that may be twisted or coiled in nature also under the microscope. And since it's gram positive, usually it will be blue in color. Okay, so you can compare the streptococcus just like this necklace that is shown here. Okay, such a blue colored round beaded necklace. It will be very much similar to that. All right. And these streptococci are like part of the normal flora in humans and animals. They are present in our body normally itself. And the first person to isolate this streptococci was Billroth in 1874. And this streptococci was isolated from acute abscesses by oxygen in 1881. And Streptococcus pyogenes, the term and that species of bacteria is isolated by Rosenbach in 1884. Okay, just for the sake of your information, know these. Okay, who isolated and what, like that. Okay. Now, next, coming to the classification of streptococcus, how you can classify streptococci. Okay, like there are like 2 to 3 classifications of streptococci, but the one that is most important is given by this Brown et al. In 1990. Okay, so this Brown et al. In 1990, he classified the streptococci based upon their hemolytic properties. All right. So hemolytic property means the ability of this streptococci to go and destroy RBCs. All right. So based upon that nature or based upon that property, Brown et al. Had like cultivated or cultured the streptococci in 5% host blood agar, and he observed the results. Okay. So when he observed the result, he was able to find 3 types of streptococci. Okay. The first one is the alpha hemolytic streptococci. Okay. So this is how the alpha hemolytic streptococci will appear. Okay. It will produce like a greenish discoloration in the petri dish. Okay. Like a very greenish discoloration. Why this green discoloration? Because this group of streptococci is not able to completely hemolyze the RBCs. Okay, they were not able to like completely destroy the RBCs. Only partial hemolysis is happening there. Okay, so that's why it appears greenish in color. And some of the most common ones in this alpha group is the viridans streptococci. Okay, the Streptococcus viridans and the Streptococcus pneumoniae. All right, so these are the 2 most common streptococci that is present in the alpha group. Then we have the beta hemolytic streptococci. In beta hemolytic streptococci, it will appear something like this. Okay, completely colorless. Completely colorless. Why it is colorless? Because the streptococci will go and destroy all the RBCs. Okay, complete hemolysis happens. That's why it's colorless. Okay, so the main organisms that are involved in this beta hemolytic streptococci is Streptococcus pyogenes and Streptococcus agalactiae. Okay, pyogenes is an important streptococci, medically very important streptococci. And then the last group, that is the gamma hemolytic streptococci. Okay, so in this gamma hemolytic streptococci, what happens is that no hemolysis happens. That is, it will appear reddish in color itself. Okay, not reddish in color. Yes, that's the color of the host blood agar, but there will be not much changes. Okay, so this is how it will appear in gamma hemolytic streptococci. The streptococci will not be able to lyse any of the RBCs. Okay, so it will just take the color of the host blood agar. All right, so it will appear like this. And the some of the important streptococci that is coming under this gamma hemolytic streptococci is the Enterococcus faecalis. Okay, the all those streptococci are included under gamma hemolytic streptococci. So this is the major classification of streptococcus given by Brown et al. Based upon the ability, based upon its hemolytic properties. Okay, hemolytic properties means based upon its ability to go and destroy RBCs. Okay, so based on that, there are three groups of streptococcus: alpha, beta, and gamma. All right. Then there is another classification also based upon the carbohydrate antigen. Okay. So in around 1933, this Lansfield, he like classified this streptococci based upon the nature of carbohydrate antigen that is present on the cell wall. Okay. So this streptococcus has cell walls, and the cell wall has lots of antigens. Now, there is a particular antigen that is called as the carbohydrate antigen. So, based upon the presence of this carbohydrate antigen, Lansfield has like classified this streptococci into like around 20 groups. Okay, so these 20 groups are named alphabetically from A to H and K to V. All right, there is no I and J, but all the other alphabets are included. Okay, A to H and K to V. Okay, so this group A, okay, this group A of this Lansfield group contains mainly the Streptococcus pyogenes, and this Streptococcus pyogenes is again subclassified into different types based upon Griffith type A. Okay, so Griffith had also subclassified this Streptococcus pyogenes groups. Then next is based upon the biochemical test. Okay, based upon the biochemical reactions. Okay, like catalase positive or hydrolase positive or oxidase positive. Based upon that reaction, the viridans group of streptococci. The viridans group of streptococci mainly includes Streptococcus mutans, mitis, salivarius, and swankis. Okay, so they are classified based upon this biochemical test. Then based on the oxygen requirement, also Streptococcus had been classified. We have the facultative anaerobes. Facultative anaerobes means they can grow with or without oxygen. Okay, in the absence of oxygen also, they can thrive. Okay, somewhat facultative means flexible, flexible in nature. Okay, they can grow with or without oxygen. Flexible in nature, that's why FF. Okay, facultative, flexible. Then we have the obligate anaerobes. Obligate anaerobes include species like Peptostreptococcus. Okay, they like hate oxygen. Okay, they do not need oxygen at all. They can only thrive without oxygen. Okay, so such species include the Peptostreptococcus species. Okay, one example is the Peptostreptococcus species. And other example of this facultative anaerobe include Streptococcus pyogenes. All right, so these are regarding the classification of Streptococcus. So usually the essay question usually start like classify Streptococcus. Okay, so when you get an essay question like this, first write a short introduction about streptococci and then move on to the classification. In the classification, give major importance to the hemolytic groups. Okay, alpha, beta, and gamma, and then the Lansfield groups also. All right. Okay, so let's have a look at some of the medically important streptococci. Okay, so pyogenes is the most important one, and this bacteria is usually present in the throat and skin. It can cause like upper respiratory infections, pyoderma, rheumatic fever, glomerulonephritis is all caused by this Streptococcus pyogenes. Then we have Agalactiae, which can result in meningitis, neonatal meningitis, and septicemia. All those diseases can be caused by Agalactiae. Then we have Equisimilis, which causes usually the pharyngitis. Streptococcal pharyngitis is also an important topic, and it can also cause endocarditis. Then anginosis, which causes usually pyogenic infections. Okay, like pus-forming infections can be caused by this sign, anginosis. Then fecalis, which is usually present in the colon, which is mainly responsible for UTI. and endocarditis and other suppurative infections. Then bovis is also present, which is usually indicated in causing endocarditis. Then viridans streptococci. Okay, so this viridans group, it contains the sanguis, mutants, mitis, salivarius, and all. Mutants, the most common one and the important one, which is causing dental caries. Okay, that's why the streptococci group becomes very important from the dental perspective. All right. Then we have the sanguis, which is mainly responsible for endocarditis. Okay, so these are some of the medically important streptococci. Please go through that. If they had mentioned some of the medically important streptococci and the diseases caused by them, you can include those tables. Then, how this streptococci is causing these diseases? Okay, so this streptococci, they usually cause the diseases with the help of their toxins. Okay, so there are so many toxins that are released by streptococci, but let's discuss some of the most important ones. The first and the most important one is hemolysins. Okay, just like the name suggests, hemolysins. Okay, they are the go and destroy RBCs. That's why they are called as hemolysins. Now, these hemolysins are of two types. We have streptolysin O and streptolysin S. Okay, O refers to oxygen labile. Okay, labile means unstable. Okay, it will not be stable in the presence of oxygen. Okay, so that's called as streptolysin O. Then we have streptolysin S also. The streptolysin S means it is oxygen stable and it is also soluble in serum. Okay, that's why it's streptolysin S. Okay, so the streptolysin O, it is usually oxygen level, heat level, cardiotoxic, and leukotoxic in nature. And the streptolysin S, it is oxygen stable, soluble in serum, and it is just a protein. Okay, it is not antigen. Okay, an antigenic protein will be capable of producing antibodies. Okay, but this streptolysin S, it is just like a normal protein. Okay, it will not elicit any immune response. Okay, so that's about the hemolysins. Then we have the streptococcal pyogenic exotoxin. Okay, so this streptococcal pyogenic exotoxin is also called as erythrogenic toxin or dick toxin or scarlatinal toxin. Okay, so why it is called as erythrogenic or scarlatinal type of toxin? Because when this toxin is injected into like susceptible individuals, they were producing an erythematous reaction. Okay, like a reddish area is produced in the skin. Okay, so that's why it's called as erythrogenic toxin. Now, there are three types of the streptococcal pyrogenic toxin, type A, B, and C. This A and C are coded for by the bacteriophage, while B is usually by the chromosome. Okay, so that's about streptococcal pyrogenic exotoxin. Then we have streptokinase. Streptokinase is also called as fibrinolysin. Fibrinolysin means it has the capability to this enzyme. This particular enzyme has the capability to lyse the fibrin. Okay, to break off the fiber. Okay, so where this will like help the streptococci for spreading the lesion or for spreading the infection. Okay, so when this enzyme is released, it will go to the surrounding of the infection and will start degrading the fibrin around. Okay, where is fibrin present? Fibrin is present in the connective tissue. Okay, so it will like lyse all the fibrins and it will help in the spread of the infection. Okay, so it helps in breaking down the fibrin barrier around the lesions and it also facilitates the spread of infection. Now, this is an antigenic protein. Antigenic protein means this is a protein molecule that is capable of triggering an immune response. That is, because of this particular enzyme, the antibodies can be produced against it. Okay, so that's about streptokinase. Then we have DNase or the deoxyribonucleases. Deoxyribonucleases will help in the depolymerization of the DNA. Okay, so most of the infections of the streptococci, if you look at it, they are pyogenic or suppurative lesions. Okay, so this suppurative lesions means they are pus forming lesions. Okay, so once the bacteria goes and attack the tissue, the pus will be formed, and this pus will be containing lots of necrotic cells. Okay, the cells are destroyed and it is like becoming pus. So it will contain lots of necrotic cells. These necrotic cells will contain lots of DNA. All right. So this DNA needs to be cleaved or needs to be broken down. Okay, so that the pus will become a little bit, what to say, thinner. Okay, so the pus can become a little bit thinner. Okay, so for that, this DNase enzyme is mainly helpful. So this DNase enzyme, it helps in the depolymerization of DNA, and it also helps in liquefying the thick pus. Okay, so if the pus is like too thick, then it cannot drain outside. Okay, so to liquefy the pus also, we are this DNase enzyme will help. There are 4 types of DNase enzyme. We have the A, B, C, and D, and B is the most antigenic type. All right. Then we have the NADase, or the nicotinamide adenine dinucleotidase enzyme. It is antigenic and it is also leukotoxic in nature. Okay, so it has like uh, toxicity against the leukocytes. Then we have the hyaluronidase, which have the same function like the fibrinolysin. It breaks down the hyaluronic acid. Okay, hyaluronic acid is mostly seen in the basement membranes and in the connective tissue. So when this enzyme goes and breaks down the hyaluronic acid, it helps in spreading of that particular infection. Okay, so this is also antigenic in nature. Then we have the serum opacity factor. The serum opacity factor mainly includes lipoproteinases. Okay, so this lipoproteinases determines how much virulent a streptococci should be. Okay, so these lipoproteinase only determines how much virulent a particular streptococci must be. Okay, so it determines the virulence of the organism. Then other enzymes like proteinases, phosphatase, esterases, amylase are also present, which mainly helps in the spread of the infection and causing symptoms for the patient. All right. So this is about the toxins that are present in the streptococci. Now coming to the diseases that are caused by streptococcus. Okay, so these streptococcus pyogenes mainly cause 2 types of diseases. We have the suppurative diseases and non-suppurative diseases. Suppurative diseases means those are pus-forming lesions. Okay, pus will be formed in those diseases. That's why it's called as suppurative diseases. Then we have the non-suppurative diseases also. So these suppurative diseases can be seen in, since this streptococci is present in almost all parts of the body, it can be causing infection in any part of the body also. Respiratory infections will happen, which is most common, which includes like tonsillitis and streptococcal pharyngitis. Then skin and soft tissue infections can happen, like lymphangitis, cellulitis, erysipelas, or the impetigo can result, or necrotizing fasciitis. Okay, necrotizing fasciitis is usually caused by a mixed anaerobic and aerobic bacteria, and that is usually called as flesh-eating bacteria. Okay. Then the toxic shock syndrome, genital infections, all can be included under these suppurative diseases. Then other than that, we have the non-suppurative diseases also. Okay, so these non-suppurative diseases are extremely important because usually in the essay question or as a short note, they can ask like, write a note on the non-suppurative diseases of streptococcal diseases. Okay, so there are usually 2 non-suppurative diseases caused by the Streptococcus pyogenes. One is acute rheumatic fever. Okay, one is acute rheumatic fever and the other one is post-streptococcal glomerulonephritis. Okay, so one is affecting the heart and the other one is affecting the kidneys. Okay, that is the acute rheumatic fever and the post-streptococcal glomerulonephritis. Why they are called as non-suppurative diseases? Because they do not result in pus formation. Okay, so these 2 diseases, no pus is formed. Whether all these diseases, yes, These are suppurative lesions, pus is formed. But these two, last two, no pus is formed. That's why they are called as non-suppurative diseases. So let's have a look at in the essay question, it was given like write note on the pathogenesis of the streptococcal pharyngitis. So let's see how this streptococcus is causing this pharyngitis. So Streptococcus pyogenes is the causative organism for the streptococcal pharyngitis, and it is a normal commensal in the respiratory tract and in the throat area. So this Streptococcus pyogenes, it will go and attack the epithelial cells in the throat. Okay, so how it attacks the epithelial cells in the throat? There is a particular membrane for the Streptococcus, which is called as the lipoticoic acid. Okay, so this lipoticoic acid, okay, the outer layer is a capsule and the fimbriae and all are present. But just below this capsule, you have this protein and lipoticoic acid layer. Now, this lipoticoic acid layer is the major help or major friend for this Streptococcus pyogenes, which will help in attaching this bacteria to the pharyngeal epithelium. Okay, so when this bacteria goes and attacks the pharyngeal epithelium, there is a particular receptor in the epithelium also, which will like readily receive this bacteria. Okay, so what is that receptor? That receptor is the fibronectin. Okay, so fibronectin is present on the epithelial cell, which will readily receive this bacteria. And in the bacteria, there is this lipoticoic acid layer and this fimbriae. It will help in readily going and attaching with these epithelial cells that are present in the pharynx. Okay, so the help is from both sides. Okay, the bacteria is also doing its part and our body is also doing its part. Okay, so it will come and attack the pharyngeal epithelium, and initially it can cause tonsillitis, and diffuse spread can result in pharyngitis, and if it is still untreated, it can lead to like complications like otitis media, mastoiditis, quinsy. Okay, quinsy, Ludwig's angina, life-threatening. Okay, Ludwig's angina is a life-threatening condition and it's an emergency condition. Okay, then the suppurative adenitis also. Okay, so such complications can also arise because of this streptococcal pharyngitis. Okay, so this is the basic pathogenesis behind streptococcal pharyngitis. Now let's have a look at the non-suppurative streptococcal diseases. Okay, so this non-suppurative streptococcal diseases, like I had mentioned, it mainly includes two diseases. One is acute rheumatic fever and other one is the post-streptococcal glomerulonephritis. Okay, so in this acute rheumatic fever, when does it occur is that if a patient is attacked by this streptococcus. Okay, so the patient has a streptococcal infection. Now, after 3 to 4 weeks of this streptococcal infection, the patient can develop carditis. Okay, inflammation of the muscles of the heart. There will be connective tissue degeneration of the heart walls. There will be inflammatory myocardial lesions like Ascoff nodules. Okay, Ascoff nodules are like a pathognomic feature of rheumatic fever. Okay, they are like small inflammatory lesions that are present in the muscle of the heart. Okay, so these Ascoff nodules can be present, and these are all formed as a result of some kind of hypersensitivity reaction. Okay, so already infected streptococcal patient, after like 3 to 4 weeks, maybe because of the diffuse spread of the lesion, what happens is that these bacteria can go and infect the heart muscles. Okay, the walls and the connective tissue of the heart vessels can be destroyed because of these vessels, and it will result in something called as acute rheumatic fever. All right, now this is one non-suppurative lesion. Okay, it is a not non-pus forming lesion. Okay, then the next one is post-streptococcal glomerulonephritis. Okay, so this post-streptococcal glomerulonephritis is again the patient is affected with a streptococcal infection after like 1 to 2 weeks. The patient may or may not be affected with the streptococcal infection, but usually after 1 to 2 weeks, what happens is that that patient will have like a sudden like a damage to the kidney function. Okay, so this sudden damage to the kidney function following a streptococcal infection, that is called as post streptococcal glomerulonephritis, just like the name. Okay, post streptococcal. Okay, after the streptococcal infection, some kind of a inflammation of the kidneys are happening. Okay, so this is mainly caused due to type 3 hypersensitivity reaction, and the main bacteria that is involved in this post streptococcal glomerulonephritis is group A beta hemolytic streptococci. Okay, so this group A beta hemolytic streptococci mainly includes Streptococcus pyogenes, but not all strains of Streptococcus pyogenes are capable of producing this glomerulonephritis. There is some special strains are involved. They are called as the nephrogenic streptococci. Okay, so these bacteria that is mainly involved in this post-streptococcal glomerulonephritis, they are called as nephrogenic streptococci. Okay, compared to acute rheumatic fever, the prognosis is much better for this post-streptococcal glomerulonephritis, and it is a self-limiting disease also. Self-limiting means during the course of time, it will heal on its own. Okay, so that's called as the self-limiting disease. Now, let's like compare between these two non-suppurative streptococcal diseases, the acute rheumatic fever and the post-streptococcal glomerulonephritis. Acute rheumatic fever, the primary site of infection is usually the throat, whereas for GMN, it is usually the throat or skin can be affected. Okay, prior sensitization, that is, like I had mentioned, a patient had streptococcal infection. Okay, that was the primary infection. After 3 to 4 weeks only, these non-suppurative diseases are getting caused. Okay, so this is called as prior sensitization. Already the patient is affected with streptococcus. So to that patients only, mostly these non-suppurative diseases are occurring. Okay, so this prior sensitization is very much essential in case of ARF, whereas it is not that necessary in case of GMN. All right. Then any type of streptococcus species can cause this acute rheumatic fever, but the main streptococcus pyogenes strain, like the ones that are mentioned here, only these strains can cause GMN, glomerulonephritis. Okay. The immune response will be very marked in case of ARF, where it is comparatively moderate. Okay. And the complement system is also somewhat unaffected in case of ARF, but the complement system is lowered in case of GMN. Okay, what do you mean by complement system? Complement system is the group of blood proteins that help the immune system in fighting the infection. Okay, so they are like the friend of the immune system. Okay, complement system is just like the friend of the immune system. It helps the immune system in fighting the infection and for clearing the damaged cells and all. Okay, so this complement system is usually unaffected in case of ARF, where it is very much lower in case of GMN. Okay, and genetic susceptibility may be present for ARF patients, where it is not that much research for glomerulonephritis patients. And repeated attacks are very common in case of ARF, where it is absent in case of GMN. And penicillin prophylaxis. Okay, penicillin prophylaxis means giving penicillin antibiotic as a preventive measure. Okay, that is essential for an ARF patient because it is an acute case. Okay, whereas it is not indicated for a streptococcal glomerulonephritis. The course of this ARF is usually progressive. Okay, initially it will be like low, or it can be progressive in nature. Okay, the condition usually worsens over time. Okay, but in case of GMN, it usually spontaneously resolve. That is, it can heal on its own. Okay, so because of that, the prognosis of the disease is very good for GMN, whereas it is comparatively variable for the ARF. Okay, so this is the main comparison table between the non-suppurative streptococcal diseases. Non-suppurative streptococcal diseases are very, very important. Please have a look at it before you go and attend your exam. Okay. Now coming to the laboratory diagnosis, the most important part in microbiology, always any bacteria they ask. Okay, even in short notes, they can ask like laboratory diagnosis of mycobacterium, laboratory diagnosis of staphylococcus, laboratory diagnosis of streptococcus, like that. Laboratory diagnosis is an important topic in microbiology. Please study it thoroughly. Okay, so in the laboratory diagnosis, usually the streptococcal specimens are taken from the throat swab, or from the, if it's a skin, pus swab, or in case of any exudates, we are taking that exudate as the sample. Okay, now how we can transport this sample to the lab? With the help of Pike's media. Okay, so this Pike's media is the transport media for this Staphylococcus aureus. This Pike's media, it usually contain saline or phosphate buffer and some kind of proteins like peptone or serum, and reducing agents, and the base, that is the agar or gel, will be there. Okay, only usually the basic constituents are only present in this Pike's media. Okay, so once it reaches the laboratory, you can make a smear. Okay, you can make a smear of it and stain it and observe it under the microscope. Now, once you observe it under the microscope, what you will find? You will find gram-positive cocci. Gram-positive cocci means, in the first slide I showed you, like the blue roundish cocci. Yes, those will be seen, which will be usually spherical or oval in shape and very small. Okay, like only 0.5 to 1 micrometer in diameter, and they will be usually arranged in chains also. Okay, just like a necklace chain, it will be arranged like that also. Okay, and you can also culture this specimen. Okay, so culturing, once you culture this, what happens is that it can be cultured in blood agar media, or it can be cultured in liquid media, or it can be cultured in any other selective media also. Okay, so once it cultured this blood agar, you'll be able to see small, circular, semi-transparent, convex disc like this. Okay, this yellowish colored small areas that you see in this picture, yes, those are all streptococcus colonies. Okay, and the virulent strains, okay, like the one that has the capability to cause disease, they will be usually appearing mated in appearance, somewhat dull, mated in appearance, whereas the avirulent strains, okay, the ones that are not capable of causing any disease, they will be appearing glossy in nature. Okay, under this blood agar. Then we can use the liquid media also. For the liquid media, you can choose either glucose or serum broth, and it will produce something like a granular turbidity. Okay, granular turbidity will be formed in the liquid media. Okay, so as a storage medium, you can use the Robertson's cooked meat medium also. Okay, then you have the selective media also. Usually, the Streptococcus pyogenes, it will be very resistant to crystal violet. Okay, more than any other bacteria, this Streptococcus pyogenes will be very much resistant to this crystal violet. So if you're using this crystal violet, it usually will not grow in that media. Okay, so the selective media can be like a crystal violet can be used. Then coming to serology. Serology means we are looking at the antigen levels and the antibody levels. Okay, based upon the antibody levels, we can predict whether, okay, this person had streptococcal disease or not. Okay, so the most important method that we use is anti-streptolysin O, or ASO titration. Okay, ASO titration is one of the most common methods that we use for prior streptococcal infection to check whether there was streptococcal infection in that particular patient. Okay, so if the ASO titer levels are greater than 200, then it is confirmed that the patient had prior streptococcal infection. So such high levels will be usually seen in acute rheumatic fever patients. Okay, so all the other methods that we had mentioned, the specimen microscopy and the culture methods, are usually used for suppurative lesions. Okay, for non-suppurative lesions, we usually take these serology tests. Okay, acute rheumatic fever and glomerulonephritis, we usually take this serology test. Then next is the anti-deoxyribonuclease B. Okay, so this anti-DNase enzymes or DNase B titers will also be greater than 300. If it is greater than 300, it shows that significant infection has happened. And this is like streptococcal pyoderma, this anti-deoxyribonuclease B levels will be very much higher. Okay, then we have the streptozyme test also. The streptozyme test will be like positive for all these streptococcus throat and skin infections. Okay, so this is also an important test which will help us in analyzing whether the patient was infected with streptococcus or not. Okay, so this is about the laboratory diagnosis of streptococci. Very, very important. Next, you have the anaerobic streptococci. Okay, whatever we had discussed till now, the Streptococcus pyogenes, mutants, the Mitis salivarius, and all the other lesions that is causing the particular diseases are all aerobic streptococci. Okay, but there is a particular category of streptococci which are called as anaerobic streptococci also. This anaerobic streptococci alone can be asked as a short note question, or it can be included under the part of the essay question. Okay, so these anaerobic streptococci mainly includes Peptostreptococcus. Okay, so this Peptostreptococcus is the major anaerobic streptococci. The species that are included under this Peptostreptococcus include anaerobius and stomatis species. Okay, they are the major ones that are included under this Peptostreptococcus. But other than that, there will be fine Goldia magna and Peptonophilus acycloreticus and Anirococcus prevotei and Anirococcus tetradius can also be present. Okay, so these are all very small gram-positive cocci. They are just like 0.3 to 1 micrometer in size, and they are arranged in chains, just like the streptococci. And since they are anaerobic, usually this carbohydrate fermentation that takes place with the production of acid or gas, or like both are produced. Okay, they are non-spore forming, non-motile, anaerobic, and catalyzed negative, just like all the other streptococci. The comparatively all the features are the same, but just the difference is that they are anaerobic in nature. Okay, so these anaerobic streptococci are also present in the normal flora of our oral cavity, GIT, genitourinary tract, skin, and all. Okay, so what all diseases this anaerobic streptococci can cause is in the brain, it can cause brain abscess. Okay, in the respiratory tract, it can cause lung abscess, empyema, bronchiectasis, or aspiration pneumonia. Okay, in the female genital tract, it can cause sepsis and tubo-ovarian abscess, Bartholin's absence, septic abortion, wound infection following any genital surgery are all caused by this Peptostreptococcus. In the skin and tissue, it can cause infection of the sebaceous cyst, axillary abscesses, breast abscesses, and cellulitis, diabetic ulcer, gangrene can all be involved with this anaerobic infection. Okay, most of the lesions are, yes, abscess and suppurative in nature. Yes, most of these suppurative lesions can be caused by this anaerobic streptococci. Okay, so this is all about the streptococci. We had discussed about the classification, the various types of streptococci, the medically important ones, and the laboratory diagnosis and the diseases caused by them, and the non-suppurative streptococcal lesions, anaerobic streptococci, what are they? Like that, we had discussed almost everything in detail. So if a question on streptococci is asked, please write according to what is asked in the question. Okay, based upon that, please include whatever is necessary. Okay. Now, next is malaria. Okay, so for in the past, so many question papers I had seen this malaria being asked frequently, the causative agents of the malaria and the life cycle of Plasmodium and laboratory diagnosis of Plasmodium. They were like commonly asked. So this malaria, it is a protozoal disease. Okay, it is caused by a protozoan. Okay, which protozoan is responsible for this malaria? It is Plasmodium. Okay, or Plasmodia. Now there are 4 types of Plasmodia which can cause malaria. One is Plasmodium vivax, second one is Plasmodium malariae, third one is Plasmodium ovale, and fourth one is Plasmodium falciparum. Okay, so these four like protozoans are responsible for causing malaria. Now, out of these four, the first three, okay, vivax, malaria, and ovale, they cause benign type of malaria. Okay, benign type of malaria means it will be somewhat self-limiting. We can control it with the drugs, and the patient comorbidity and mortality rates are comparatively less. Okay, it is not that much of a serious condition. Okay, so that is called as benign malaria. Okay, but the last one, this Plasmodia falciparum, okay, it causes the most serious, or what to say, the most dangerous form of malaria, that is called as the malignant malaria. Okay, so this Plasmodium falciparum is the one that is causing malignant malaria. Okay, so how a human can get malaria? With the bite of a female Anopheles mosquito. Okay, so this is a most common disease in the tropical areas, like in the Africas, in parts of South and Central America, in India and Southeast Asia and all. And usually this malaria does not affect sickle cell patients. Okay, why? Because this Plasmodia has an affinity for RBCs. Okay, healthy RBCs. Okay, so if the RBC is unhealthy or sickled, then these protozoans cannot go and infect and like live inside them. Okay, so that's why this malaria is comparatively less in case of sickle cell patients. Now let's have a look at the life cycle of Plasmodia. Okay, so this life cycle of Plasmodia is completed with the help of two hosts. Okay, we have the mosquito and the human. Okay, so with the help of these two hosts only, the life cycle of Plasmodia is getting completed. In the mosquito, the this particular Plasmodia will undergo its sexual cycle. Okay, it will undergo its sexual cycle and will produce sporogone. Okay, in the humans, it takes the asexual cycle. Okay, which is called as the schizogone. Okay, so in humans, normally liver can be affected and all the RBCs can be affected. Okay, so there is a liver stage and an RBC stage. Okay, so this liver stage is called as the exoerythrocytic stage, and the RBC stage, it is called as the erythrocytic stage. Okay, so the life cycle of RBC is completed in mosquito and in humans. Okay, both mosquitoes and humans help in the completion of this life cycle of Plasmodium. Okay, so in the mosquito, sexual cycle happens. In the humans, asexual cycle happens. Okay, so let's discuss in detail what is happening. Okay, so if the mosquito is going and biting an infected person, okay, so the mosquito is going and biting an already infected malaria patient. Okay, so what happens is that in the blood of that infected patient, there will be gametocytes present. Okay, gametocytes means there will be a male and a female protozoan will be present. Okay, so this mosquito will take this male and female protozoans inside its body. Now, once this male and female gametocytes enter inside the mosquito body, where it will go? It will go straight to the gut. Okay, in the gut of the mosquito, these female and male protozoans will mate with each other and they will form, like after 10 to 20 days, they will form sporozoite. Okay, these terms are really important. They will form sporozoite. Okay, when they form this sporozoite, these sporoforms will be released and they will go and reside in the salivary glands of the mosquito. Okay, so gametocyte or the mosquito is going and biting an already infected malarial patient. In the blood of the malaria patient, there will be gametocytes present. These gametocytes will go inside the mosquito they are male and female gametes. Okay, so they will mate with each other at the gut of the mosquito, and after like 10 to 20 days, it will form sporozoids. Now these sporozoids will go and resides in the salivary glands of the mosquito. Okay, so once they reside in the salivary gland of the mosquito, this mosquito is now infected. Okay, so this infected mosquito, if it goes and bites a normal person, okay, it is going and biting a normal person. Now, when it goes and bites a normal person, what happens is that in the saliva, there will be sporozoids. Okay, so these sporozoids will be released into the blood of the human. Okay, so these sporozoids are released into the blood of the human, and these sporozoids will directly go to the human's liver. Okay, it will directly go to the human's liver, and in the liver, it will form something called as hepatic schizont. Okay, what do you mean by schizont? Schizont is usually like a mother cell. Okay, this mother cell will have like lots of baby parasites. Okay, so that is called as hepatic schizont. Now, this schizont is formed inside the liver. Now, inside the liver, this schizont will multiply and form lots of merozoites. Okay, so schizont is the mother cell, whereas these merozoites are the daughter cells. Okay, so the schizont will multiply, 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 and it will form lots of merozoites. Now, these merozoites will go and attack RBCs of the blood. Okay, so when it attacks RBCs of the blood, a part of the merozoites will become gametocytes. Okay, so a part of the merozoites, it will become gametocytes, and other remaining merozoites, it can form like trophozoites. Okay, trophozoites are nothing but they are the ring form that is appearing inside the RBCs in malaria patients. Okay, they will be appearing like a ring inside the RBC of the malaria infected patients. I will show you how it appears. And these merozoites, what happens is that it will also form like schizont, other mother cells also, and it will again form lots of merozoites, and like that it is continuing as a cycle. Now these RBCs, it can rupture. Okay, once it ruptures, what happens is that these merozoites will be released to the blood, and it will cause the clinical signs and symptoms of malaria. Okay, so when these RBCs rupture, again, these merozoites will, these gametocytes will be present in the blood, and again, the cycle is continuing. Okay, another mosquito comes and bites this infected individual. It is going as a cycle again and again. Okay, so this is regarding the life cycle of Plasmodia. Try to study it like a story. It will be much easier for you. Normal mosquito, infected person, gametocytes in the mosquito, it will go to the gut. The gametes will mate and it will form sporozoites. The sporozoites will be seen in the salivary gland of the mosquito. From the salivary gland, it will go and bite a normal person, and these sporozoites will be transferred to the blood of the normal person. It will go straight to the liver. The liver, there is a schizont stage. Schizont stage, it is like a parent cell. Okay, so it will form many daughter cells like the merozoites. These merozoites will go and attack the RBCs. In the RBCs, there will be trophozoid stage. Okay, it will form like a ring structures will be formed in the RBCs, and the gametocytes are also formed at the same time. And these merozoites will get converted to schizont, and it will produce more and more merozoites, and the RBC will rupture and release all these merozoites into the blood, and it can cause the diseases, all the signs and symptoms of malaria. And again, if an mosquito is coming and biting this infected person, again, the cycle is continuing again. Okay, so this picture here shows the clear life cycle of Plasmodia. So remember these flow charts. Okay, this will be much easier for you to remember. And like I had told earlier, this Plasmodia falciparum causes malignant type of malaria. Okay, that is very serious or very endangering. Okay, it causes increases mortality rate of individuals. Okay, it is a very dangerous form of malaria. Okay, so how this Plasmodium falciparum alone is different from the other species? Okay, what makes this Plasmodium falciparum different from the other species that is causing this malignant type of malaria? Okay, that's because the first reason is there is no exoerythrocytic stage. No exoerythrocytic stage means in the humans, once it enters, this sporozoites enters inside the human, there are two stages. One is exoerythrocytic stage and other one is the erythrocytic stage. Erythrocytic stage means the changes that are happening inside the RBCs. And the exoerythrocytic stage means the changes that are happening inside the liver. Okay, so there is no liver stage in this Plasmodia falciparum. Okay, these porozoites are directly going and attacking the RBCs. That's why it's more dangerous. Okay, and any erythrocyte can get infected. Okay, so other species like the ovale or the vivax and the malariae, other species, they usually target only the juvenile RBCs or the young RBCs. But Plasmodium falciparum is not like that. It can target any RBC. Okay, there is no specificity. Any erythrocyte can be affected. And usually, other Plasmodia, if there is one RBC, only one parasite will go and infect these RBC. Okay, but Plasmodium falciparum is not like that. If there is one RBC, like multiple Plasmodium falciparum can go and infect it. Okay, so that's the third reason. And fourth reason is that once infected, these red cells, they become sticky in nature. Okay, they become very sticky in nature and it can clump and cause obstruction of the blood vessels. Okay, so that can result in ischemia formation and microinfarcts and all those things can be formed. Okay, so that can make things much more dangerous. Okay, so these four are the prime reasons why this Plasmodium falciparum is causing malignant type of malaria. Okay, and what are the clinical features of malaria? There will be fever and it will be cyclic in nature. Okay, cyclic peaks of high fever, then chills will be there, anemia can be there, splenomegaly can be there. Anemia because like lots of RBCs are getting destroyed. So yes, anemia can be there. And these destroyed RBCs are going to the spleen. So yes, hepatosplenomegaly can also be there. This malaria can affect, malarial parasites can affect any part of the human body. Okay, in the brain, it can cause encephalopathy. Okay, it can cause something like cerebral malaria. Okay, the characteristic feature of the cerebral malaria is granulomas. Okay, not just the bacteria are capable of forming granulomas, but protozoans like this Plasmodium can also form granulomas. Okay, that too inside the brain. Okay, so this is a classical example of granuloma that is formed inside a brain of the malaria infected patient. Okay, then heart can also be affected, leading to congestive heart failure. Liver can be affected, hepatomegaly, spleen can be affected, splenomegaly, kidneys can be affected, which will result in hemoglobinuria nephrosis. Okay, so when the kidneys are affected, it will result in like very dark colored urine. Okay, which is also called as color colored urine. Okay, so why this color colored urine? Because lots of RBCs are getting destroyed. Okay, so because of this intravascular hemolysis, what happens is that there will be hemoglobinuria. Okay, so these RBCs will be excreted out of the body through the urine. Okay, so the urine will appear dark black or like color colored urine. Okay, and that is called as black water fever. Okay, so that is called as black water fever. And usually this black water fever is seen in the Plasmodia falciparum affected patients. Okay, and towards the, if you look at the complications of this malaria, the patient can go into coma or hypoglycemia, renal impairment, severe anemia can happen, hemoglobinuria, jaundice, pulmonary edema, acidosis, congestive heart failure, and hypotensive shock can all result from malaria. Okay, so it is a life-threatening disease. And how you can diagnose the malaria? The gold standard is smear. Okay, take the blood of the patient and make a smear. Okay, if you are making a thick smear, you will be able to see the parasites. Okay, if you are like able to make a very thin smear, then you can even identify the parasite, whether it is vivax or whether it is oval or whether it is malaria or whether it is falciparum. That can be detected with the help of thin smears. Okay, and then there are rapid diagnostic tests also. We have HRP2 and PLDH. So this HRP2 is nothing but it is the histidine-rich protein 2 for diagnosing mainly the Plasmodium falciparum, and PLDH is parasite lactate dehydrogenase. It can detect all the other species of Plasmodium. All right, then serology test. Okay, antibody titers. Look for the antibodies. Then fluorescent dyes can also help in the recognition of uh, Plasmodium species. So this is the smear that is taken from a malaria patient. You can see the RBCs. Okay, these small cells that are seen here, they are the RBCs. And look at these RBCs. There are some small yellow dot-like structures that you see, which are in a like a ring shape. Yes, this is the ring stage or the trophozoid stage of Plasmodium is shown here. Okay, so this is regarding the malaria laboratory diagnosis. So I think I had covered all the info that is related to malaria, its types, the causative agents, and what kind of malaria it is forming, and the life cycle. Okay, life cycle is frequently asked, and also the laboratory diagnosis of malaria. Okay, so that's all covered. Now we are moving on to the basics of microbiology, that is the culture media. Okay, culture media is one of the frequently asked questions. Sometimes they can ask the whole culture media and classify them like that, or they can ask like a particular culture media, like write about enrichment media, or write about selective media, or write about transport media. Like that also, specific individual culture media is also can be uh, included. Okay, so this, what is this culture media? So this culture media is more like a soil to the plant. Okay, so a plant needs a good soil for it to grow, right? So just like that, the culture media is like the soil for the bacteria. Okay, it's like a food for the bacteria. Only if the culture media is good, then the bacteria will grow. Okay, otherwise the bacteria will not grow. Okay, so this is about the culture media. It is like a nutrient rich substance that is used to grow, maintain, and study the microorganism in a laboratory. Okay, basically it is like the food for the microbes. If the food is good, the bacteria will grow. If the food is not good, the bacteria will not grow. Okay, so this culture media can be like liquid or solid in nature. Okay, that is one classification. Okay, we have the liquid culture media and the solid culture media. Liquid culture media includes like the brain, heart, infusion growth, peptone water, nutrient growth are all included under this liquid culture media. The bacteria that we isolate from the blood or water can be easily grown in this liquid culture media. But the bacteria that is grown in this liquid media will be usually diffuse in nature. Okay, so we cannot study the characteristics of the bacteria that clearly. Okay, so for that, what we do is that we can use solid culture media. Okay, so the solid culture media includes the mainly the base will be the agar. Okay, it can be nutrient agar or blood agar or chocolate agar can be used. So in the, if you are using a solid culture media in the petri dish, we can actually see the growth of the bacterial colonies, how they are growing, and what are the characteristic features it shows. Like that, all can be studied very clearly. Okay. And other than this liquid and solid culture media, there is another classification that is called as the simple, complex, and synthetic or defined. Okay. Simple means it is the the culture media that we use for growing the bacteria is very simplest. Okay, that is not too much like what complex ingredients will not be present. Okay, it will be a very simple dish. Okay, it will be like a very simple dish or a simple food for the bacteria. Okay, the one common example is the nutrient broth. Okay, the peptone, meat extract, and sodium chloride and water. These are the only ingredients in this nutrient broth. So this nutrient broth comes under this simple culture media. Then we have the complex culture media. Okay, complex culture media means to the base, we are adding like lots of ingredients for the specific growth of specific bacteria. Okay, so this complex culture media, it includes thiosulfate, citrate, bile salt, sucrose, agar, like that are all included under complex culture media. Okay, the dish is more complex. Okay, then we have the synthetic or defined culture media. Synthetic or defined culture media means it is made up of pure chemical substance. Okay, no natural substance is used here, only pure chemical substances are used here. Okay, so it includes Hank's balanced salt solution. Okay, so this is one way to classify the culture media. Can be liquid and solid, or it can be simple, complex, or synthetic or defined. Okay, and the next classification is based upon their functional requirement. Okay, so based on the functional requirement, the culture media can be classified as enriched, enrichment, selective, indicator, differential, and transport media. Okay, so there is, you can remember an easy mnemonic to remember the different classes of culture media. You can remember it by elephants, E for elephants, E for eat, S for sweet, I for icy, D for delicious, and T for treats. Okay, so elephants, eat, sweet, icy, delicious, treats. Okay, you can remember it like that, or you can make your own mnemonic also, no issues. Okay, so this enriched media is the one to which we are adding some kind of nutritions. Okay, so the base of the culture media will be always that is called as the basal media. Okay, so the basal media is usually made with only agar. Okay, so to this agar, if we add nutrients only, the bacteria will grow. Okay, so what kind of nutrients can be added? Blood can be added, egg can be added, and serum can be also added. Okay, so this will enrich the media. Okay, so such kind of culture media are called as the enriched media. So this enriched media includes blood agar and chocolate agar. Now, there is something called as enrichment media. Okay, so what do you mean by enrichment media is you are taking the saliva sample or a swab from the oral cavity. Okay, and you want to study just the Streptococcus mutans. Okay, but in the oral cavity, there will be lots of other bacteria also. There will be Streptococcus mutans, there will be Streptococcus sanguis, there will be Streptococcus mitis, there will be like lactobacilli will be present, Truvotella, like that. All those species will be present in the oral cavity. But we just need Streptococci. Okay, so what you can do is that you can use the enrichment media. So what this enrichment media does is that it will suppress all the other commensal bacteria. Okay, so all the other bacteria other than the streptococci is suppressed. Okay, so when other bacteria are suppressed, only the streptococci will be remaining back. Okay, so that is called as enrichment media. And one example is the tetrathionate growth and the selenite F growth. Okay. Then we have the selective media. Selective media is, again, you are taking a saliva sample. Okay. You only want Streptococcus mutans. You don't want other bacteria. Okay. So what you can do is that you can give this selective media to that petri dish. Okay. When you give the selective media to the petri dish, what happens is that only the Streptococcus mutans will grow. Okay. The enrichment media is suppressing the other commensal bacteria, whereas the selective media is, it is selectively growing the desired bacteria. Okay, only the required bacteria will be grown in greater numbers. Okay, we are particularly selecting that bacteria. Okay, so that is called as selective culture media. And the main examples are desoxycholate citrate agar and thiosulfate citrate bile sucrose. Okay, so these are all selective media. Next, we have the indicator media. Okay, indicator media means, again, if we want to culture only Streptococcus mutans from the saliva sample. Okay, so what we can do is that we can add an indicator to that particular petri dish. Okay, so when you add that indicator, what happens is that when the mutants are growing, that colonies will change color. Okay, so when we see the change in color, we'll be able to know that, okay, so these bacteria are going inside this petri dish. Okay, so that is called as the indicator media. If you are culturing the bacteria in an indicator media, it will change color. Usually, when uh, uh, for example, the Wilson Blair medium. Okay, in the Wilson Blair medium, it will be usually colorless, but when this Salmonella typhi is getting grown in this Wilson Blair medium, it will produce black colored colonies. Okay, so when you see this black color itself, you can predict that, okay, so the typhi colonies are growing here. Okay, so that is called as this indicator medium. Then you have the differential media also. Differential medium is mainly used for differentiating between two species. Okay, you have the lots of Streptococcus species inside the oral cavity. You want to differentiate between the Streptococcus salivarius and Streptococcus mutans. Okay, so that for that you can add some kind of differential media. Now, when this differential media is used, the mutants colonies will be in one color and the salivarius colonies can be in another color. Okay, for example, I am telling this, it can be differentiated like this with the help of differential media. Okay, so the commonly used differential medias are McConkey medium and Naegler's medium. McConkey's medium, it contains like peptose, lactose, agar and all, neutral red and all. And it is used to differentiate between lactose fermentase and non-lactose fermentase. Okay, these lactose fermenters will produce pink colored colonies in the McConkey medium, whereas the non-lactose fermenters will be usually colorless or pale in the McConkey medium. Okay, so that's how you differentiate between uh, this lactose fermenters and non-lactose fermenters with the help of this McConkey medium. Then we have this transport medium also. Transport medium means to carry the specimen from the clinic to the laboratory. Okay, you need a transport medium. Okay, this transport medium will withstand drying of the medium, and it will like preserve the medium in its own natural condition. Okay, so the usually used transport medium is Stewart's medium. Okay, Stewart's medium is like a non-nutrient. It is a very soft agar gel, and it contains usually some kind of reducing agent to prevent oxidation. And it may also contain charcoal to neutralize all the bacterial inhibitors. And okay, so that's about this Stewart's media. Okay, then other than this media, there is something called as anaerobic media also. In this anaerobic media, it usually includes Robertson's cooked meat medium and thioglycolite medium. Okay, so these anaerobic media are specifically used for growing anaerobic organisms alone. Okay, mainly used for anaerobic organisms. It includes Robertson's cooked meat medium and thioglycolite medium. All right. So that's about culture media. Now we are going on to Vincent's angina. Okay, Vincent's angina is very important. You will study about it in detail in fourth year perio. So this Vincent's angina, it is also called as necrotizing ulcerative gingivitis. The patient will clinically appear like this. It is also called as trench mouth. Okay, why trench mouth? Because this disease was very common during the First World War among the soldiers. Okay, because of the stress and because of the other conditions. The most of the soldiers who are participating in the World War were affected by this Vincent's angina or this necrotizing ulcerative gingivitis. Or that's why it's also called as trench mouth. Okay, so yes, it is a very painful condition. It is the gingiva will become inflamed like this and edematous like this. Swelling of the gingiva will be seen, and you see bleeding from this interdental papilla area. Yes, these are all characteristic features of this Vincent's angina. Like the characteristic lines that is used to describe this Vincent's angina is punched out crater-like necrosis will be seen. Okay, so this is the punched out crater-like. Crater means like a depression. Okay, depression like necrosis is seen, and it will be usually covered by a grayish pseudo membrane also. Okay, so when you remove the pseudo membrane, you can see bleeding also. Okay, and it will produce a very fetid odor, a bad smell, a pain will be there, spontaneous hemorrhage. Okay, just touch the gingiva and it will bleed readily. Okay, and lots of accumulation of necrotic debris can also be seen. Okay, so this is about Vincent's angina. This Vincent's angina was actually described by John Hyacinth Vincent, and he suggested that this Vincent angina is caused due to Fusobacterium and spirochetes. Okay, both the Fusobacterium and spirochetes are involved in the etiology of Vincent's angina. So he used the term like fusospiroketosis. Okay, both the fusobacteria and spirochetes are involved. Okay, so which fusiform bacteria is involved? Fusobacterium nucleatum and Borrelia vincenti. Okay, so Fusobacterium nucleatum and Borrelia vincenti are the major causative organisms for this Vincent's angina. Now, other than that, Leptotrichia bacillus, which is also called as Vincent's fusiform bacillus or Fusobacterium fusiforme, which is an anaerobic gram-negative bacilli. Okay, they are also involved in this pathogenesis of Vincent's angina. Okay, so Borrelia vincenti, it is a gram-negative motile spirochetes. You can see the Borrelia vincenti here. Okay, so this is how, if you take a sample from the Vincent's angina patient, this is how it will appear. Okay, lots of spirochetes will be there, and in between, you will be able to see fusobacteria also. Okay, so that's why it's called as fusospiroketosis. Both the fusobacteria and the spirochetes are involved in the pathogenesis of Vincent's angina. All right. Now we are moving on to inclusion bodies. Okay, so where will you see in these inclusion bodies? These inclusion bodies are mainly seen in the viruses. So when a cell is attacked by virus, what all changes can happen? Acantholysis can happen. Acantholysis means destruction of the cell can happen. Nuclear clearing. Nuclear clearing means if this is a cell and this is the nucleus. Now, surrounding the nucleus, you will be able to see a very small hollow space. It is called as nuclear clearing. Okay. Then nuclear fragmentation. Yes, sometimes the nucleus can get fragmented and will produce like fragments of the nucleus can also be seen. Or sometimes the nuclear enlargement can happen. Okay, the ballooning degeneration. Okay, nucleus can become very big also, depending upon the virus which attacked it. Okay, and sometimes what happens is that inside these cells, you can see inclusion bodies. Okay, so what are these inclusion bodies? Inclusion bodies are nothing but they are like the aggregates of proteins that are formed because of this infection from virus. Okay, so such type of bodies are called as the inclusion bodies. Now, these inclusion bodies are the characteristic hallmark of a disease. Based upon the type of inclusion body that is present inside the cell, we can predict which virus has attacked this particular individual. Okay, so these inclusion bodies are the typical hallmark of the disease. Okay, so these inclusion bodies can be present inside the cytoplasm or they can be present inside the nucleus. Now let's see what these viral inclusion bodies are. Inside the cytoplasm, you have these inclusion bodies, Henderson-Patterson bodies that are present in molluscum contagiosum, Negri bodies that are present in rabies. Okay, rabies can be asked as a short note question. When asked as a short note question, please mention about this inclusion bodies also. Then Guarneri bodies, Passion bodies, Bollinger bodies, Borel bodies are all seen in this intracytoplasmic inclusion bodies. Then we have the intranuclear inclusion bodies also. Intranuclear inclusion bodies can be acidophilic or pinkish in color, or they can be basophilic or bluish in color. Okay, so the acidophilic intranuclear inclusion bodies include Cowdery type A and type B. So this Cowdery type A inclusion bodies are usually seen in varicella zoster virus infection. Okay, and in herpes simplex and in yellow fever virus. Okay, whereas Cowdery type B, which is acidophilic in nature, is seen in poliovirus, and type B, which is basophilic in nature, mostly bluish in color, is seen in adenovirus and cytomegalovirus. Okay, so herpes simplex infections like Cowdery type A are like really common. You can see the inclusion bodies in here. Okay, intranuclear inclusion bodies can be seen here. Okay, and here also. The Cowdery type A and type B inclusion bodies are seen here. Okay, so these inclusion bodies are caused as a result of attack by the viruses. Inclusion bodies can be asked as a short note question, or it can be asked along with the essay question also. Okay, now which sections you should concentrate more on before going for your exams. Okay, so while I was going through the question paper, more and more of essay questions, okay, essay questions and short note questions were asked from bacteriology topic. Okay, which all bacteria you should focus on, I'll tell you, but bacteriology is extremely important. Then it is followed by virology. Okay, which are viruses you should study, that also I'll tell you. Then the next majority of questions were asked from immunology. Okay, the hypersensitivity reactions, the autoimmune diseases, are everything can be asked from immunology. I'll also discuss with that. Then mycology and parasitology and general microbiology. General microbiology, I'm mainly including the sterilization and all those things. Okay, so under the bacteriology chapter, please learn about mycobacteria. Okay, mycobacteria is a frequently repeated essay question, very frequently repeated. Mycobacteria, about the features of the tuberculosis, about BCG vaccine, about national the program, okay, the vaccination program and all, TB elimination program and all. Okay, there was one question in which they had asked about this NTEP program also. Okay, so you should have a look at that. Mycobacteria and TB, really, really important. This is a topic that you are studying in general pathology also. So while studying, study it combinedly. Okay, look at the clinical features and the pathogenesis from the pathology part, and the laboratory diagnosis and all those things from the microbiology. Okay, so when you are learning it as a whole, and the whole topic is covered at one go. Okay, so try doing that. Then Clostridia, gas gangrene, another favorite topic of examiners. The laboratory diagnosis of Clostridia about the gas gangrene are very commonly asked. Conibacterium diphtheriae, the diphtheria disease, the DPT vaccine is commonly asked. Spirochetes, okay, syphilis. Syphilis is very, very commonly repeated question. Okay, the test for the syphilis also, that's also important. Okay, then the staphylococcus. Okay, staphylococcus laboratory diagnosis and the lesions produced by the staphylococcus. Again, streptococci also we had discussed in detail about streptococci. Then Clostridium tetani and the tetanus infection and the prophylaxis that you can give for this tetanus infection, all are important. And anaerobic bacteria and its culture methods are also important. Okay, so essay questions were asked a lot from bacteriology. So please try to focus on these bacteria. Other than that, the methicillin resistant staph aureus, anaerobic culture methods, bacterial growth curve, structure of the bacterial cell, the Vincent's angina, which we had discussed today, culture media, which also we had discussed today, viral test, bacterial capsule, Weil's disease, viral test conjugation, and all can be asked from this bacteriology topic. Okay. Now, coming to virology, the most important one is definitely HIV, the morphology of HIV, the opportunistic infections associated with HIV are all important. Then poliovirus. Okay, the prophylaxis for poliovirus, rabies virus, and hepatitis B and hepatitis C viruses and its serological markers. Okay, then measles, mumps, rubella, MMR, and its vaccines also, MMR vaccine also. Related to that, the Koplik spots and inclusion bodies can also be asked. And oncogenic viruses. Oncogenic viruses, you will have an idea about it if you learn in the neoplasia in general pathology. Have a look at the oncogenic viruses. Then COVID virus. Okay, it's COVID virus after the COVID period. Yes, the questions on COVID virus can also be asked. Okay, so have a look at the coronavirus also. Okay, so these are some of the important questions from virology. Now, coming to mycology and parasitology, candidiasis, very important. Non-albicans candida also. Okay, that is not just Candida albicans. There are other species also, like Candida glabrata and all other species also there. You should know their names and the diseases caused by them too. Okay, then parasites, malarial parasites, the classification, the life cycle of the plasmodia, which we have discussed today, can be asked. Intestinal amoebiasis. Okay, amoebiasis is an important question. Rhinosporidiasis, then culture media for the fungi, for intestinal nematodes. Okay, hookworm, tapeworm, like that. For intestinal nematodes also, you have to have an note on it. Black water fever. Okay, black water fever, which is seen in the malarial infection. We had discussed it today. Entamoeba histolytica, amoebiasis again, and oral lesions of systemic mycosis. Okay, so these are the types by which these mycology questions can appear for your examination. Okay. Other than that, discuss in coming to the immunology, autoimmunity and autoimmune diseases, very important. Hypersensitivity reactions, extremely important. Antigen antibody reactions. Okay. What are the antigen antibody reaction? And you have to know about ELISA, Western blotting techniques, like that, the techniques also. Okay. Have a look at the PCR, ELISA, and Western blotting techniques. Okay. Then immunoglobulins, the types of immunoglobulins, its uses, its structure, very, very important. Agglutination reactions and four agglutination tests, active immunity, passive immunity, acquired immunity, innate immunity, serum sickness, heterophile antigens, super antigens are all important topics from immunology. Now, if you look at the general microbiology part and the medical microbiology part, sterilization is another favorite topic. The hot air oven, moist or dry heat method, autoclave, and chemical sterilization methods in hospitals. Those are all like frequently asked. Then contributions of Louis Pasteur. Okay, they can sometimes ask short notes like that also. You should know the contribution of Louis Pasteur. National immunization schedule. Okay, and standard precautions against bloodborne infections, mutational drug resistance. Okay, just T lymphocytes alone can be asked as a short note. Culture media. Okay, either together or like separate, like the selective media can also be asked. Okay, mechanism of antibiotic resistance and its prevention. Okay, so these all can be asked in the as short notes. Okay, short notes are the sections where you can expect like lots of surprise questions. So yes, any topic related to these topics can be asked. Okay, it's not necessarily repeated, but yes, some of the questions are repeated again and again. So please have a look at it. Okay, so I think that's like too much microbiology for today. So if you have any doubts, you can ask me now. You can put it under the chat box, or you can put it under the comment section under this video. We will definitely answer all your queries. or you can like put it in the WhatsApp groups. We'll definitely answer all your queries, and or you can directly DM me also. Okay. So that's all for today. All the best for your exam preparations. Do well, revise, and read well. Okay. So all the best. Good luck. Good night and goodbye. Bye.